Hi everyone. Today we shall have a brief overview of energy metabolism pathways. When we look at various metabolic pathways in biochemistry, sometimes it gets very confusing.
To make sense of it all, we need to have an integrated approach. We don't want to get lost in the bizarre networks of chemical reactions after all. So, here we go.
By the end of this lecture we shall know about the energy metabolism pathways, simplified view of well-fed and starvation state, and, finally, the hormonal regulation of these pathways.
Let's get started then. We all know that to carry out physiological, metabolic, or any function we utilize ATP as our energy currency. ATP, on hydrolysis, generates energy which we utilize for our bodily functions. And these are the sources that we utilize to make ATP: carbohydrates, lipids, proteins, and ketone bodies. This is where things start getting confusing.
We have several metabolic pathways related to energy generation and storage. There are glycolysis, Krebs cycle, fatty acid oxidation, gluconeogenesis, fatty acid and triglyceride synthesis, ketone bodies synthesis and utilization, and other associated pathways. These pathways must be tightly regulated. We do not want energy to be wasted when there is plenty. Neither do we want insufficient energy to be produced when required. How this balance is created, is what we are going to discuss next.
Which pathway is predominant depends on the state we are in, and on the dominant hormonal influence in each of these states. Let's broadly divide these states as well-fed state and starvation state. There are again subdivisions of starvation state, but for the sake of simplicity, let's go with this classification. Glucose is the predominant energy source during well-fed state, whereas fats and ketone bodies are the predominant source of energy during starvation state. There are certain exceptions and modifications to them. For example, red blood cells will always utilize glucose as energy source irrespective of the state. We shall certainly discuss them later, but for a while let's hang on with our assumptions.
Let's just write them down, these two states. Well-fed or right after meal state on the left-hand side, and the starvation or in between meals state on the right. The predominant source of energy in the first state is glucose, and the major source of energy in the starvation state is fats and ketone bodies. We shall talk about the role of proteins in a while. Also note that there are exceptions to these, some of which will be dealt right in this lecture. It's the right time now to talk about the hormonal influences. The most important hormone secreted during the well-fed state is insulin. After meal, the plasma levels of glucose rise drastically. The rise in glucose is the most important stimulator for insulin release by the pancreatic beta cells. You can say, in the well-fed state, we are under the spell of insulin. Meanwhile, during the starvation state, the insulin levels drop. Instead, another hormone secreted by the alpha cells of pancreas rises, the glucagon. Two other hormones rise as well, besides glucagon: the epinephrine, by adrenal medulla, and the cortisol, by adrenal cortex. The balancing of all the pathways we listed earlier is majorly controlled by these hormones. Let's just memorize them again before we discuss the actual balancing act. In the well-fed state, the insulin is the hormone. In between meals, the trio of glucagon, epinephrine, and cortisol are the hormones. Now that you know it, let us discuss each of these states, and the respective hormonal influences separately. Let's start with the well-fed state. Remember, we are under the insulin influence right now. The primary thing to do after any state is to fulfill our energy requirement. That is what every cells do. Most cells primarily utilize glucose to make ATPs in this state. Let's take an example of liver here. In a well-fed state, it takes glucose, convert it into pyruvate in the cytoplasm, and then to acetyl coenzyme A in the mitochondria. Through substrate-level phosphorylation that happens during glycolysis, and oxidative phosphorylation that happens through the electron transport chain, finally, the ATP is formed. The same thing happens in all the tissues: brain, skeletal muscles, adipocytes, etc. Red blood cells also utilize glucose, but unfortunately, it cannot make ATP from aerobic glycolysis, Krebs cycle, and oxidative phosphorylation due to the lack of mitochondria. The only ATP source for RBC is substrate-level phosphorylation during anaerobic glycolysis. Remember, the end product of anaerobic glycolysis is lactate, which needs to be recycled, and will be discussed later during starvation. The heart muscles primarily feed on fatty acids even during the well-fed state rather than glucose. During starvation, they prefer ketone bodies, along with fatty acids, which we shall discuss in the subsequent section. Besides these modifications and exceptions, the major way to generate ATP is almost same for most tissues. Aerobic glycolysis, followed by Krebs cycle, electron transport chain, and finally, the oxidative phosphorylation. Now, after the energy requirements are satisfied, it is sensible to store the glucose for future requirements. In the liver itself, the extra glucose molecules start polymerizing to form glycogen. The process is called glycogenesis. Similar thing happens in skeletal muscles as well. The only difference is, the glycogen formed by the muscles can be utilized only by themselves during the starvation state, but the glycogen formed in liver can provide glucose to other tissues as well. While it was indeed a good idea to store glucose in the form of glycogen for future requirements, as it happens, we can only store a very limited amount of glycogen in the liver. This would provide sufficient glucose to the other needy tissues for probably 12 hours or so, but not much more than that. We need alternative ways to store energy during the well-fed state. Acetyl coenzyme will come to the rescue here. We could not store glycogen because they were osmotic molecules. Not as much as monomeric glucose, but still osmotic. But, if we could polymerize acetyl coenzyme A, it would form fatty acids. First, palmitic acid, and then several other types. Then, 3 fatty acid molecules would combine with a glycerol 3-phosphate to form triglycerides. Triglycerides are simple lipids that are completely insoluble in water. We can store them as much as we like, as, unlike glycogen, they would not draw water. So, glucose from diet is utilized for 3 major processes: making ATP, storage as glycogen, and finally fats for longer storage. But, fats should not be stored in liver. Too much fat in the liver is an initial sign of an injured and unhealthy liver. Therefore, what it does is, make triglycerides, add a bit of proteins, called ApoB100, and form a complex lipid called VLDL, a short for very low-density lipoproteins. This VLDL is then exported to the blood, where it matures and finally delivers triglycerides to the perfect storage tissue, the adipocytes. How it does so is the topic of lipoprotein metabolism, so let's not go too deep there. But, let it suffice to say that the extra glucose left after ATP generation and glycogen storage, in the well-fed state, is finally converted to fats in the liver, and then delivered to the adipose tissues for storage. During starvation, many tissues will feed on these fats, which is a very efficient source of energy. Much more than carbohydrates. We do have another source of fat as well. The fat from diets. For transport into the blood, they undergo similar strategy as VLDL. They combine triglycerides, cholesterol and phospholipids together, then associate itself with apoproteins called ApoB48, and form a lipoprotein just like VLDL. Only this time they are called chylomicrons. Chylomicrons formed in the intestine finally enter the circulation via lymphatic ducts and undergo the similar fate as VLDL. Which is to say, they too deliver their triglyceride components to adipose tissues. Again, the details of this process are a bit out of scope for this discussion, but, I hope you get that, the lipids from our diet form chylomicrons in the intestine, whereas, the glucose from our diet forms VLDL in the liver. After entering the circulation, they serve a similar purpose: to deliver fats into the adipose tissue for storage. This sums up the major processes that occur during the well-fed state. To summarize, In this state we are under the insulin influence. Besides generating energy, the extra food sources are stored for future use. Glucose is the major fuel here. Some glucose in our diet are converted to its polymer glycogen in the liver and skeletal muscles. The remaining glucose are converted into fats in the liver, and finally delivered to the adipose tissues for storage. Similarly, the lipids in our diets are also exported to the adipocytes for storage. Also, do remember the exception. Let's discuss the starvation state now. Since there is no external food supply, it is now time to utilize the food sources that we stored during the well-fed state. Remember, we are primarily under the influence of three hormones during starvation: glucagon, epinephrine, and cortisol. The major energy sources are fatty acids and ketone bodies. But, some organs will still prefer, or even, are dependent on glucose for energy. Let's discuss them one by one. In the post-absorptive state, multiple pathways are going on simultaneously. The whole logic behind these pathways is simple though. It is all about what is the primary source of energy to which tissue. Let's start with liver and adipocytes. When the insulin level is low, the adipocytes start hydrolyzing their fats into fatty acids and glycerol. These fatty acids will serve as the energy source for many tissues, including adipocytes itself and liver. The process is called beta oxidation, with the end product again being acetyl coenzyme A. Acetyl coenzyme A is literally the central molecule in the energy metabolism pathways in both states. This acetyl coenzyme A from fatty acid oxidation can then either enter TCA cycle to derive energy through electron transport chain and oxidative phosphorylation. Or, in case of liver, it can be utilized for synthesizing ketone bodies as we shall see shortly. Now, let's add a bit of a detail to this process. How do the adipocytes figure out when to store the fatty acids as triglycerides, and when to mobilize the fatty acids for its oxidation? While you must have already guessed the hormonal influences, now, let's discuss something about the two enzymes that these hormones regulate. Both are lipases by nature, which means they break down lipids. While one is found on the external surface of endothelial cells in close proximity to adipocytes and other peripheral tissues, the other is found inside the adipocytes. Let's discuss briefly about these two lipases. The first lipase is called lipoprotein lipase, or LPL for short. They are expressed by endothelial cells of the capillaries near the peripheral tissues such as heart, muscles, or adipocytes, as shown here. They break down the triglycerides from chylomicrons and VLDL during the well-fed state. And yes, insulin does activate this enzyme, as you must have already guessed. This is what the LPL do. Hydrolyze TGs into fatty acids and glycerol during well-fed state, so that the fatty acids can be taken up by adipose tissues for storage, so that it can be utilized later during the starvation state. What about the glycerol part? It is taken up by liver to make TGs from fatty acids. The adipose tissues cannot convert glycerol to glycerol 3-phosphate, which is required for TG synthesis. But, liver can easily do so, because they have the enzyme called glycerol kinase that the adipocytes lack. If you are thinking, the next lipase must then be active in the starvation state, you are absolutely correct. This lipase is called hormone-sensitive lipase, or HSL for short. They are named so because they are very sensitive to low levels of insulin, which is what happens during the starvation state. They may also be sensitive to high epinephrine levels, but the major sensitizer is still the lack of insulin. What happens is pretty simple. During starvation state, HSLs inside the adipocytes are activated due to low insulin levels. They hydrolyze the TGs that are inside the adipose tissues, converting them to fatty acids and glycerol, and dump them into the circulation. Since they are water-insoluble, fatty acids are carried by the major transport protein of the plasma, the albumin. They then enter various tissues like heart, liver, muscles, etc. for oxidation, generating plenty of energy. Even the adipose tissues themselves now feed on fatty acids. The end product, acetyl coenzyme A, can enter the Krebs cycle to generate further energy. Meanwhile, in the liver, the acetyl coenzyme A will make another energy source, called ketone bodies. This is what we shall discuss now. Ketone bodies are another good fuel during starvation state. There are 3 ketone bodies that can do so: acetone, beta-hydroxybutyrate, and acetoacetate. All of them are derived from acetyl coenzyme A, produced during fatty acid oxidation. They are synthesized in liver cells after their energy demand are met. Unfortunately, the organ that makes ketone bodies cannot utilize it as ATP source. This is because the hepatocytes lack an enzyme called thiopherase. Many extrahepatic tissues meanwhile, devour ketone bodies as fuel. Muscles, especially the heart muscles, love ketone bodies. Kidneys are another example. Ketone bodies produced by liver are dumped into the blood. They then enter these tissues, and will be converted back to acetyl coenzyme A, to derive energy. Even the brain tissues during prolonged starvation utilize ketone bodies for survival, when glucose is too low. Let's think for a while here. Aren't the ketone bodies in blood the basis for ketoacidosis? Not exactly. The ketone bodies are quickly taken up from the blood by ketone-utilizing tissues. Only in diabetics, or during prolonged starvation, this becomes a real problem. On both cases, ketone bodies are produced in much higher amount than they can be utilized, causing increased levels in blood, and hence ketoacidosis. In a normal healthy person, ketone bodies are delicious fuels for the tissues, rather than an alarming signal. As emphasized previously, there are tissues that will feed on glucose even during the starvation state. RBCs, for example, do not have mitochondria to undergo beta-oxidation of fatty acids to use it as energy fuel. Similarly, blood-brain barrier prevents fatty acid albumin complex to enter brain tissues for it to be utilized. For the sake of such tissues, somehow glucose needs to be synthesized even when there is no exogenous source. There are 2 ways to derive glucose. The easiest and quickest way is the glucose molecules themselves. Remember? We stored glycogen during the well-fed state in liver and muscles. Glycogen is nothing but a polymer of glucose. What we need now is just break them down to derive glucose. This process is called glycogenolysis. And guess which hormone will promote this process? Of course, glucagon. So, here is what happens. The liver starts depolymerizing glycogen and release glucose into blood, which is then utilized by glucose-dependent tissues like RBCs and brain. But remember, before the hepatocytes break down glycogen, they need to be satisfied by their energy levels themselves, or else it would not make any sense. Nobody donates their energy source when they themselves are hungry. The liver undergoes beta-oxidation to feed itself, and breaks down glycogen to donate it to glucose-dependent tissues. The muscle tissues have different story to tell. While they store glycogen during well-fed state, they do not donate glucose to blood for other tissues to feed on. They can feed on fatty acids, and even ketone bodies, but still they will not dump their glucose to blood. Perhaps that is why they are sometimes regarded as selfish tissues, but I wouldn't go that far. They simply lack an enzyme to convert glucose 6-phosphate to glucose. This reaction occurs during the last step of glycogenolysis. The enzyme is called glucose 6-phosphatase, and glucose in its phosphorylated form cannot escape the muscle tissues. We cannot call some tissues selfish because they lack an enzyme, can we? Let's visualize the whole scenario again. Glucose during post-absorptive state is initially derived from glycogenolysis from liver. RBCs and brain will continue with their glucose-eating habits and liver will help them do so. But, there's a limit to this. Which brings us to another way of deriving glucose, the gluconeogenesis. While glycogenolysis provide the immediate source of glucose, to fulfill the demand of glucose-dependent tissues for longer, we need other sources. These sources are not carbohydrate in nature. The process of deriving glucose from non-carbohydrate sources is called gluconeogenesis. While it starts late than glycogen breakdown, it really lasts for much longer. The process mainly occurs in liver. Some of it occurs in kidney as well. Any non-carbohydrates that can be either converted to glycolytic intermediates, pyruvate, or Krebs cycle intermediates, can act as the precursors for gluconeogenesis. For example, glucogenic amino acids. Proteins break down during starvation releasing amino acids, some of which can be converted to glucose, and are termed glucogenic amino acids. Alanine is a typical example of such amino acid. It can be easily converted to pyruvate by the help of an enzyme called alanine transaminase, or ALT for short. Pyruvate then can easily be converted to glucose in an almost reversal phenomenon. Remember though, it is not an exact reversal of glycolysis. Let's see what else can be converted to pyruvate. Remember anaerobic glycolysis in red blood cells? RBCs dump their lactate to blood, which is then taken up by liver. The lactate is then converted to pyruvate by the enzyme lactate dehydrogenase, and then back to glucose again. This glucose can be utilized by RBCs to produce lactate. This amazing cycle of interconversion of glucose to lactate, and back to glucose, is known as Core's cycle. Similar phenomenon occurs in skeletal muscles during strenuous exercise. Let's see if there is any source that can be converted to glycolytic intermediate. Glycerol, the product of fat hydrolysis, can be converted to dihydroxyacetone phosphate, an intermediate of glycolysis. This can again be reverted back to produce glucose. Hence, glycerol is also a good source of glucose, as fats are being continually hydrolyzed during starvation. What about the other product of fat hydrolysis, the fatty acids? Can they be converted to glucose? The answer is no. We'll see why. Remember the end product of fatty acid oxidation? You are right. It's acetyl coenzyme A. Pyruvate can easily be converted to acetyl coenzyme A by pyruvate dehydrogenase, a multi-enzyme complex, but the reaction is essentially irreversible. This means, acetyl coenzyme A cannot be converted back to pyruvate. Neither do we have any mechanisms to bypass this reaction, to produce other intermediates, that can be converted to glucose. Glucose, in well-fed state can easily be converted to fatty acids, but the other way around is not possible in humans. So, always remember, while glycerol is a good source of glucose, the fatty acids cannot be converted to glucose.
Let's talk about one exception here. For even-chain fatty acids, which are most abundant, all of this is true. But, for odd-chain fatty acids, which are rare, it is not exactly true. This is because the end product of their metabolism is propionyl coenzyme A, and not acetyl coenzyme A. And, propionyl coenzyme A can form a Krebs cycle intermediate, and hence can be converted to glucose.
Let's summarize the sources for gluconeogenesis again. Glucogenic amino acids. Lactate. Glycerol. And odd-chain fatty acids. Always remember, any non-carbohydrates that can be converted to pyruvate, glycolytic intermediates, or Krebs cycle intermediates can serve as a precursor for gluconeogenesis. Let's summarize the starvation state. In this state, we are under the influence of three hormones: glucagon, epinephrine, and cortisol. The major food sources are fatty acids and ketone bodies. But for certain tissues like red blood cells, glucose is the only source, just like in well-fed state. Brains feed on glucose for a long time. Only during prolonged starvation, when glucose becomes too scarce, they start feeding on ketone bodies. These are the major metabolic processes going on: fatty acid oxidation, ketone body synthesis and utilization, glycogenolysis and gluconeogenesis, and glycolysis in certain tissues. And of course, Krebs cycle, electron transport chain and oxidative phosphorylation to generate ATPs, like in well-fed state. I hope you can draw this diagram by now. When insulins are low, adipocytes hydrolyze their fats by the help of hormone-sensitive lipase. Fatty acids produced as a result is utilized by adipose tissues, liver, muscles, and many tissues for energy. The process is beta oxidation. In the liver, the end product of beta oxidation, the acetyl coenzyme A, also produces ketone bodies, yet another fuel source that can be utilized by various tissues. Liver itself cannot utilize ketone bodies though. In the ketone-utilizing tissues, they are again converted to acetyl coenzyme A, to derive ATP via Krebs cycle and oxidative phosphorylation. Some tissues like RBCs and brain, still prefer glucose as energy source. To provide glucose, liver cells undergo two processes. The first and immediate one is called glycogenolysis. The stored glycogen are broken down, releasing glucose to plasma, providing glucose for these tissues. In the second process called gluconeogenesis, glucose molecules are derived by liver from non-carbohydrate sources, like glucogenic amino acids via protein breakdown, glycerol, from fat hydrolysis, lactate, via anaerobic glycolysis, and odd-chain fatty acid metabolism. This concludes our session on starvation state. I hope the overview was helpful. Before starting to learn the details of each energy metabolism pathway, it is always helpful to get an idea about the overall strategy. Always start with the stage you are in. Then think about the hormonal influences. Is it an insulin world, or a glucagon world? Do not leave your common sense behind. There might be exceptions to simple logic sometimes, but most of the times they are helpful. For example, let's say we are on a right after meal stage. What does our common sense tell us? Make ATP first, but after your energy demands are fulfilled, store them. Right? And insulin will help do it because right after meal, we are under the influence of insulin. Revise it a few times, and I'm sure you will understand it even better. I guess that is all for today. Have a good time.